Yep, looks perfect.
Yeah, great. Thank you so much. So yeah, thank you, Summer, for this introduction, and thank you for moderating this webinar. I also want to thank Brian Pogue and the JBO editorial staff for organizing this webinar series and inviting me to be part of it. So today I want to talk to you about the feasibility of using these molecular tension probes in neurons. And before I get started, I just want to quickly acknowledge the students in my lab who have worked on this. They are listed here in reverse chronological order with the current students listed first. I also want to acknowledge students in my colleague's lab, Dr. Bonnie Feierstein, in the Department of Cell Biology and Neuroscience at Rutgers. So this has been a very collaborative project, working with labs both at Rutgers and outside Rutgers. I also want to acknowledge our funding from the NSF, the New Jersey Commission on Brain Injury Research, and internal grants from Rutgers Global and Rutgers SCORE facilities. So cells and tissues are morphologically dynamic environments. And as you can see here in this slide, you're looking at neurons extending their processes on a glass slide. And you're looking on the left at a differential interference contrast image of these neurons. And if you look very carefully, you should be able to see processes extending here. Let me see if I can point them out with my laser pointer. There's processes extending, going sideways, going up, and really very motile processes going on. On the right-hand side, you're looking at the same field of view of the cells, which have been transfected with a fusion construct, vinculin venus. Venus is a variant of the YFP protein. And what you see is that vinculin is tracking this neuronal process going down the slide and is very dynamically moving and changing its position within the process. So cellular morphology and structure determine biological function. And morphology is effectively a physical, morphological change involves physical changes that must involve forces and displacements. So one way to look at this problem is to think about it maybe in the following scheme, where in response to environmental cues, cellular forces will be created, or cells will be sensing forces, displacement will occur intracellularly, and these forces and displacement will in turn generate a signaling cascade, an intracellular signaling cascade that will activate downstream molecular sensors, which will then result in the assembly of the cytoskeleton so that cells can assume a functional morphology that is useful to their biological function. And in the case of neurons, such functional morphology could involve filopodial growth, dendritic branching, and spine maturation. And we're very interested in studying this mechanical signaling pathway, because if we do have that deeper understanding of this, we can then harness that knowledge to be able to control perhaps tissue engineering strategies in which we can control neuronal regeneration and come up with ways that could restore neuronal connections potentially after injury or after or during degenerative diseases. So the mechanical signaling pathway or mechanotransduction pathway has been put forth decades ago. I mean, it dates back to the late 70s, early 80s with pioneers like Harris and Ingber. But what we have today, I think, is perhaps some more advanced tools to be able to probe this signaling pathway. And one of the probes that I want to talk to you about today is this vinculin tension sensor, which we are trying to use in neuronal cells to see if we can study the mechanotransduction within neurons. So just a word about vinculin. Vinculin is a protein that has been shown to be involved with the adhesion of epithelial cells to the extracellular matrix, as well as the adhesion between epithelial cells at adherence junctions is very active at cell-cell adhesions. In neurons, vinculin has been less studied, but nonetheless, it's been shown to co-localize with focal adhesion kinase. It's been shown to be localized at the growth cone, or these motile regions that I've just shown you earlier. It also plays a role in neuronal filopodial development and in stabilizing dendritic filopodia and spines. And this vinculin tension probe we're going to be using has been developed in 2010 by Dr. Martin Schwartz's lab. And in this paper by Grashoff, Hoffman et al. That was published in Nature Methods at the time, you can see a depiction of it. And the vinculin tension probe basically consists of a tension sensing module, which is inserted between the vinculin head and the vinculin tail. And the tension sensing module itself is a repeated amino acid motif, GPGGA, which is derived from the flagelliform silk protein. And it's basically an elastic linker, which is inserted between two fluorophores here, M teal fluorescent protein 1 and M venus, a variant of YFP. And this fluorescent protein pair is going to act as a FRET sensor. So FRET stands for Forster or fluorescence resonance energy transfer. And it's a process by which when a donor fluorophore and an acceptor fluorophore come in close proximity to one another, when you excite the donor can non-radiatively transfer its energy to the acceptor as shown here. So when that happens, the acceptor gets excited in turn and it can fluoresce as a result of this transfer of energy. One of the conditions for this process to occur is that the fluorophores are in close proximity here, where R is on the order of 1 to 10 nanometers. So we're talking about very small molecular distances. And there should be spectral overlap between the donor emission spectrum and the acceptor excitation spectrum. And the efficiency of the process termed EFRET here is measured as the donor excitation energy transferred to the acceptor divided by the donor fluorescence in the absence of acceptor. So it's basically the amount of fluorescence quenching that the donor experiences in the presence of the acceptor divided by the fluorescence of the donor in the absence of the acceptor. So when we're looking at this tension module, just to understand a little bit how it works, if you were to apply tension on that TS module, what you would get is an increase in that distance R between the two fluorophores. And fluorescent FRET efficiency is a very strong function of R. It varies as 1 over R to the 6th power. So as you pull on that molecule and the fluorophores go further apart and R increases, you'll see a decrease in FRET efficiency as a function of force. And this calibration was undertaken when the paper was first published. And here I'm adapting a figure from that paper where you're plotting, they were plotting FRET efficiency versus force, and you can see that FRET efficiency decreases monotonically in the range of 1 to 6 piconewtons, which is the range of forces we expect to see in these molecules. So now when you insert this TS mod inside vinculin, it's going to get targeted to vinculin, and when you express this construct in cells, it's going to localize at regions where vinculin is going to be localized. And in particular here, looking at this cell, which is attached to a glass substrate, you can see that vinculin is accumulating at these areas, which are the adhesions between the cell and the substrate. And if you were to measure FRET efficiency at these locations, you could then relate that to the amount of tension the protein is experiencing at these locations, which have been segmented out here on the right-hand side in this color image. So I just wanted to say that since the very first development of these probes, this has been a very active area of research, and intracellular tension probes are being really very actively developed recently. And they've been, people have put these tension sensors in different proteins, in particular talin or cadherins and other proteins that are involved at adhesion sites between cells and extracellular matrix, as well as cell-cell adhesions. So here we wanted to really figure out if we could do these measurements in neuronal cells, because you know, a lot of this has been studied in epithelial cells, and it wasn't very clear, you know, how well we could do this in neurons. And we're very interested ourselves in the processes of dendritic branching and process extension in neurons. So we've used cortical neurons for our first studies, which are primary neurons dissected from embryonic cortex at day E18. And we've plated them on cover slips coated with poly-D, lysine, and laminin. And we've transfected these neurons early in the culture at DIV or day in vitro 4 to 6 with either one of these 3 constructs. So I've talked already about the VinTS tension probe, about the TS mod, which is the untargeted, unloaded tension probe. We're using that as one negative control, and another negative control is a tailless VinTS probe which lacks the actin binding domain. And so again, just to show you here a frame-by-frame rendition of that first movie that I had, you can again see that vinculin is expressed at the growth cones of extending neurites. You can see that it tracks the tip of the growth cone here over time, and that this is a dynamic process. So our very first measurement consisted of just collecting images of a lot of growth cones, just taking static snapshots of growth cones expressing either VinTS or VinTL. And what you're looking at here is a montage of 111 images of growth cones collected from our neurons. And on the left-hand side, you're looking at the acceptor channel signal. So that tells you the expression of VinTS in these growth cones. And on the right-hand side, a calibrated FRET efficiency measurement in these growth cones. And we've repeated that in another 100 or 99 images of neurons, growth cones expressing VinTL, again, this tailless VinTS probe that lacks the actin binding domain. And so when we looked at these, the pixels of these growth cones, if we are to plot a cumulative function distribution of all the pixels in the neurons expressing VinTS and VinTL, we find that the histogram of the VinTS expressing pixels is shifted to the left, indicating that there's some level of tension that is different in the growth cones expressing VinTS compared to VinTL, which is our negative control. And this change is very small. However, it was statistically significant. But, you know, again, this process is dynamic. And one of the issues with looking at static images is that you could be looking at a process where the growth cones are attaching and detaching as a function of time. And if you're not catching them while they're under tension, you know, that can add a lot of noise in the measurement in terms of you're really measuring both growth cones that are under tension and not under tension at the same time. So on average, it might look like one population is under tension, but perhaps we're not looking at it you know, with fine detail. So what we've begun to do recently is to look at this process in time lapse. And here's just an example of that, where you're seeing time lapse images of a growth cone taken every 10, we've taken them actually every 5 minutes, but I'm showing them every 10 minutes, where you're seeing this growth cone changing its morphology very dramatically. And when you look at the fine spatial distribution of FRET efficiency in that growth cone, you can see that it changes. You know, it's got a spatial function to it, and it's also very dramatic, meaning it starts out being at low tension with high FRET efficiency, and then it ends up going to higher tension and lower FRET efficiency. So we really need to validate these measurements and try to understand actually what may happen as a function of time. And one of the things we're also interested in is correlating the FRET efficiency measurements with the morphological change, because again, the morphological change is the physical change that's occurring, that's causing you know, these forces to occur and to change. And so we're thinking about comparing extended or flat adherent looking growth cones versus the ones that are round and not attached. And we're trying to think about relating FRET efficiency of VinTS to the circularity of these growth cones in an effort to try to correlate FRET efficiency with the physical morphological change in these neurons. So in the last few minutes here of this, I just wanted to kind of talk about the challenge of measuring these the FRET efficiency in these probes. So we've used the sensitized emission method to do this in wide field measurement. But obviously we would like to try to now make these measurements in 3D tissue models, biologically relevant 3D tissue models. And but just to explain a little bit how it works here for the sensitized emission method, and to see that it might be cumbersome for 3D measurements, what we do is we need to take measurements in 3 channels: the donor channel, the acceptor channel, and the FRET channel. And in this FRET channel is where we expect to measure that signal due to resonance energy transfer. So here we excite the sample at the excitation of the donor, and we look at the signal at an emission centered at the emission maximum of the acceptor. But again, because of the spectral overlap between the donor and the acceptor, when we do this excitation at the donor excitation, we actually see the fluorescence of the donor, and that's going to leak into our FRET channel. We also directly excite the acceptor and measure some fluorescence from that in our FRET channel. So the FRET channel has this background signal from donor and acceptor bleed through that is unrelated to resonance energy transfer that needs to be subtracted. We need to subtract that out, estimate it and subtract it out. And we furthermore need to calibrate that measurement so that we can convert that corrected FRET channel measurement to actual FRET efficiency. And this can be very cumbersome if you're doing this in 3 dimensions, needing to register all your 3 channels, do the background subtraction and all that. So we've been developing a fluorescence lifetime imaging system to measure FRET instead. And we've developed a frequency domain FLIM system to do that. And what I just want to point out here is that FRET efficiency can be rewritten as a function of the lifetime of the donor in the presence of the acceptor, tau dA, and in the absence of the acceptor, tau d. And if you have that ratio, you can get FRET efficiency. And this measurement can then be made in a single channel, only the donor channel. And you can get your FRET measurement by doing measurement in a single channel, which can greatly facilitate that. So our FLIM system was successful at measuring lifetime of the VinTS probe at the focal adhesions of epithelial cells. And we found that the lifetime measured was longer than that of the unloaded TS mod, untargeted unloaded TS mod sensor. That was very promising. Again, longer lifetime is lower FRET efficiency. And recently we have shown that a very simple measurement of phase lifetime can be used to extract an apparent FRET efficiency, which can be directly related in this linear fashion to the true FRET efficiency of the constructs that we're using. And that happens when we modulate our frequency domain FLIM system at low frequencies under about 56 megahertz. So this greatly facilitates data acquisition and interpretation in our frequency domain FLIM system. So with that, I'm just going to summarize by saying that we are developing method to calibrate and measure FRET efficiency of VinTS in neurons. Our results currently support the feasibility of making this measurement in neuronal cells and present the first assessment of this in growth cones. We're also developing FLIM FRET using frequency domain FLIM to translate the methods ultimately to 3D live tissue imaging. So in terms of future outlook for these probes, and again, the goal here is, can we test mechanotransduction in biologically relevant 3D environments? So what do we need for this? We probably need better FLIM probes that are optimized for these measurements in 3D. Perhaps we can combine FLIM with light sheet microscopy so that we can do live measurements in 3D with minimal photo bleaching. So there's some really nice FLIM cameras that are coming out on the market that are very sensitive and that can be used for wide field frequency domain FLIM. And we're also looking at the development of biologically relevant 3D tissue models that would be amenable to use with these tension probes. So we need to be able to express these tension probes in 3 dimensions in the tissues and creating, constructs where we can see fluorescence in these 3D tissues, transfect them and so forth. So with that, I just want to thank you all for your attention. And here's our contact information should you have questions following this webinar. So thanks again, everyone.
Great. Thank you, Dr. Bastani, for